let's go. First, I would like to thank this committee for the invitation to present these results here. My name is Ricardo Affeldt. I am professor and researcher at Federal University of Santa Catarina also. I will talk a little bit about some predictions we have performed with in silico models of this compound that I will call it quinoxaline only, and also the activity on inhibiting AKT1. It is a research, a project that involves different institutions and different fields of knowledge. I myself, I am a chemist. I'm not a biochemist, and I just started working with bioinformatics as well right now, which I will tell you later on. Also, I believe that everybody is aware of the problems that cancer brings us. It is the second leading cause of death. Rodrigo has already talked very well about this topic, so I will be more precise and tell us a bit more about the molecular targets we have. It is indeed a multifactorial disease. There is a genetic issue, but there is also an environmental issue, which means that as population growth, we also have the growth of the number of cancers, and of course the deaths in the world. And when we think about a molecular target for studying cancer and novel anti-cancer compounds, most part of what we see is, and what we think is about DNA molecule, which is here. We are also conducting some experiments with DNA too, but I will talk in this lecture about some classes of proteins that are involved in the cancer signaling pathway. Probably you can't see much in this picture. It is a big picture, but I would like to call your attention on this, the central role of the AKT protein in the signaling pathway of cancer. And you can also see here some keywords, some representative keywords, for instance, apoptosis, cell survival, cell cycle progression, DNA repair, metabolism, and protein synthesis, and also metastasis. So if we take a better look on the AKT process, we have here three main groups of proteins that are involved in this process that is inside the cell, inside the plasma. We have PI3K that phosphorylates AKT, and AKT is then responsible for those keywords that I have mentioned before, especially cell growth and cell survival in the cancer cases, and is strictly related to some kinds of cancers. More specifically, if we look at the protein, the AKT, it is indeed a serine threonine kinase 1 human. We have the structure, this one on the left side, where you see that there are two main domains that binds together when there is a presence of a ligand inside. So this ligand is a allosteric inhibitor that inhibits the activity of the kinase, and in the absence of this ligand, these two parts here, they split apart, they separate, they separate, and this is a crystallographic structure from PDB. And what calls our attention, or my attention more specifically, is that the structure of this ligand is usually a heterocycle, as you can see here, a benzene fused heterocycle. This is the 3D structure and also the two-dimensional structure. And we can see that the nitrogen heterocycle, it is really important because it interacts with a residue of tryptophan here by P interaction, by P stacking. So it plays a major role on stabilizing the structure of the active AKT. And with this on mind, I have to tell you, I am a chemist. I work with organic chemistry and more specifically organic synthesis. This here is a picture of my laboratory. You can see it is a very typical set of experimental chemistry. This is the laboratory of catalysis and interface phenomenon. now it's a bit weird, a picture of me here, but hopefully in the end I can explain this better. But what we have done so far, we are very interested in nitrogen heterocycles since the beginning of the group. We work a lot with these kind of compounds here that are dihydropyridines. They showed a lot of activities. We do the syntactic part, which I will tell you a little bit more. And we are not only interested on the nitrogen heterocycles, but also on the interaction of these nitrogen heterocycles, for instance, with macromolecules, which of course they're not as big as a protein itself, but they usually serve as an interesting model for us to understand these interactions between the small compounds and the higher order molecules, which you can see in these representative publications right here. And another compound that is of our interest is those ones up here. As you can see that they share a common characteristics, that is the presence of a double ring fused aromatic compounds, also with the presence of 2 nitrogen atoms in all of them. This is the basic scaffold or the main structure of the compound, which we call quinoxaline. And these compounds right here, these were not synthesized by our group, but these are you found in literature. They show some really interesting effect here as anti-8IV. And also these compounds here, they are all related to anti-cancer activity, and some of them are even in clinical trials. So it is a very interesting heterocycle. On the other hand, we are very aware that some organic compounds, and more specifically some compounds that are biologically important, they can also react in the presence of radiation, of light radiation, which is here. I have 2 main examples that I really like. One of them is the conversion of dihydrocholesterol into pre-vitamin D3, which is a very important process that only happens when we are sun exposed, for example. This is a good example. We have also a bad example. This is a thymine present in the DNA nucleoside that dimerizes into these compounds when it's irradiated with ultraviolet. And these 2 examples are very interesting because one of our major concerns in our group is to create new organic molecules or to synthesize new heterocyclic compounds, but instead of using classic chemistry, heating or this kind of stuff, we want to use light as a source of light for preparing these compounds. So this is really important. This is how we prepare the compounds that I will show here, the quinoxaline substituted derivatives. We did it in 2 steps only, in a very mild condition using blue light irradiation. First by photooxidation of an alkyne, and then by cyclization with the product from step 1. So it is a very simple and fast protocol. We can achieve a large number of compounds. I'm showing here only these ones with yields that are good and the yields that are not so good at all. But also these compounds, not only the bioactivity and the anti-tumor relation is interesting for us, but they are also fluorescent compounds. So they could be applied on bioimaging or microscopy, for example, to observe and label biostructures. And we are able in our group also to perform this characterization of fluorescence. These compounds were made by Jennifer. The year was 2020, so we were in the middle of the pandemics that affected our experiments, of course. But back at that time, we were also starting a collaboration with Professor Rosangela from the experimental biochemistry lab. And this part of the in silico predictions then were performed by her group and by Rodrigo that speaks before me. We started with the bioinformatics tools from Swiss. I believe this is a very interesting big starting point for testing our compounds. And all the results I'm going to show here are from these compounds specifically, okay? But we have done for the others as well. The first thing was to evaluate if the compound is able to permeate through brain-blood barrier. When we submit the structure in the Swiss platform, Swiss ADME, we see that our compound is right here in the middle of this yellow part. This is the boiled egg, known as boiled egg graph, that correlates the log P with topology of this compound. And we see that, yes, it is very likely that he could permeate through brain-blood barrier. We also have this bioavailability radar here, where he matches some features concerning size, polarity, solubility. We have only one violation here, which is related to the excess of unsaturations in this compound. But this is not so bad, because we can use these results to in the future prepare some new compounds that may fit inside of all this radar. Our compounds, they are considered small molecules, which is very interesting for a drug. In the same tool, we can check for the drugability and drug likenesses. We see that there are no violations concerning different methods, Lipinski, Gose, Weber, and others. So this is a very nice starting point. And if we use the same tool from the Swiss for targeting predictions, we see there is a significant number here in this chart that is related to kinesis, which is also the category of the AKT1 that I showed in the beginning. And also, if we change for the well-known platform way to drug, we also see these results where we can get nearly 300 different targets that this compound may be able to interact, also related to kinesis. So this motivated us even more. In the way to drug, we have the tool that can predict the sites of metabolism concerning the structure of the compound. If you see here, I'm showing only the cytochrome oxidase groups that are susceptible to the action of this enzyme. We have mainly the methyl groups connected to the nitrogen, and we also have the meta-aromatic groups. So, and these groups, they are mostly affected by action of cytochromes. And when we go to the prediction of activity with the Pass Online tool, those results are really they really are together, because most part of the compounds where we see activity against on some enzymes are related to oxidation and reduction processes, which is strictly related to the groups we have in the organic molecule. For instance, here NADPH, peroxidase inhibitor, phospholipase inhibitor, monoxygenase, and so on. I do not intend to discuss all the data we have achieved with the way to drug platform, but we also did a gene expression profile induced by the same compound. And what gets my attention particularly is the in silico prediction of cytotoxicity for tumor cell lines. And as you can see here, we have several types of, in this chart here, several types of cell lines, different kinds of tumor, where our compound may be active. So with these results in hand, I told you before that I am from Brazil. I am a professor in Brazil, but right now I am as invited researcher in ITQB Nova. It is an institution in Portugal. And here we are working more closely with bioinformatics, molecular docking, molecular dynamics, and these are the results that I'm going to show you right now. For the same compounds we were discussing before, first we have energy and geometry optimization using the Gaussian software with this method, DFT calculations. With this structure in hand, we have this is considered our ligand for molecular docking with its rotations on the sigma bonds. There are actually only these ones, 3. The protein is the AKT1 from the PDB for EJN. It is considered our rigid macromolecule. Of course, we have done the treatment of the PDB by removing waters, ions, and other ligands. But nevertheless, we had to model the structure because some parts of these compounds were not crystallized. So we started then with this simulation. We simulated a protein. The molecular docking was performed with the Autodoc 4.2 and Autodoc Vina and Autodoc Suite for visualization. One thing that is interesting is that the pocket from the active site is particularly big concerning if you compare this compound, the quinoxaline compound here, with the original ligand, for instance, that I showed in the beginning of the lecture. Our compound is much more smaller than the original ligand, for instance. Even though when we perform 1,000 scans in the Autodoc software, it shows a clustering energy histogram that is not actually a histogram. We have only one cluster of conformations here that fits all together in the same value of energy, of association energy between our heterocycle, the quinoxaline heterocycle, and the protein itself. So this is a really interesting result. Also, what we have done, we have and this result that I'm showing here is even with what we call a blind docking, which means that the whole protein is inside the box, and the calculation were performed with the whole protein. With Autodoc Vina, we were able to get a lower value of binding energy, which is minus 11.2 kilocalories per mole. And this value also didn't change with 3 random seeds. Here we have an image that was prepared with Pymol, and we also performed the interactions using Protein Plus. This is a tool an online tool from University of Hamburg. It's very interesting. It works in the same way as LeakPlot and Molegro as well, where you can see the interactions between the heterocyclic core with tryptophan, the same tryptophan that I mentioned in the beginning of the speech. And also we have several hydrophobic interactions. I think this picture might be a little bit better for you to see. Here is the tryptophan. Here you have the P interactions. You also have hydrogen P interactions with a tyrosine residue back there, and hydrophobic amino acids as well. So what we are doing also, we did the molecular docking with all the series of the compounds. We see pretty much the results are very similar for these compounds, which is interesting. If we change that group for another, for example, for an oxidized group, nitrosyl group, we see there are hydrogen bonds with threonine too. And what we are doing right now, our perspectives are ongoing experiments in the with collaboration of the 3 laboratories, is to perform molecular docking with other proteins, other AKTs. DNA is also something that we are doing right now with different classes of compounds, not only these ones that I showed you. Molecular dynamics using the GROMACS package, the GROMACS software, with these compounds. In vitro tests are being conducted in the experimental biochemistry lab with a pattern of several cell lines. To see if this relation that we are observing in the computational is indeed important. Inhibition of AKT kinetic experiments, they will be conducted soon, and of course this will guide us for design and synthesis of new compounds. So what I have presented right now is maybe it's not so much of a computer-aided drug design, because we have done the design prior to the calculations, but those designs of these compounds were based on the PDB structures. I would like to take some minutes to show you really quick. This is a picture from Florianópolis, a city in Santa Catarina, in the south part of Brazil. My time is almost going out. In the Federal University of Santa Catarina, this is a picture of our group. And this is where I am right now in Portugal. So you see that both cities, they have very beautiful landscapes. I would like to thank you. I would like to thank everybody that was involved, specifically the invitation to be here today. With those pictures I showed before, the photographs of me and the other scientists, I would like to remind you that science is made of people. I wanted to ask Professor Rosângela, Rodrigo, that is here too. He made all the part of In Silico. Elena, we've been working together for years. We are even married. Claudio, Gianna, Jennifer, all the students, and where the money comes from. CAP is from Brazil and FC2 in Portugal. Thank you very much for your attention.